Today, we're diving into a technology that is seriously rewriting the rules of neuroscience. We're talking about human brain organoids. We're going to look at how they're made, how they're completely changing medicine, and yeah, the really profound questions they force all of us to ask. So let's get right into it.
It sounds like something straight out of science fiction, doesn't it? But, this very question is right at the heart of one of the biggest leaps in medical research today. And the wild part is, scientists aren't just asking this question anymore. In a very real way, they're starting to answer it.
So, what are we actually talking about here? These are human brain organoids, or HBOs. Now, they're these incredible 3D structures grown from human stem cells that kind of mimic how our own brains develop. But, and this is absolutely crucial to understand, they are not mini brains. That term, while it's super catchy, is just misleading. They can't think, they can't feel, they are amazing but simplified biological models for research.
So how in the world do you grow one? Well, it all starts with something called pluripotent stem cells. These are cells that have this just amazing ability to become any type of cell in the body. Scientists give them a gentle nudge, guiding them to become neural cells. Then, and this is the incredible part, they basically step back. They let the cell's own natural programming take over, allowing them to self-organize into this complex, three-dimensional structure. It's really less about building something and more about, guiding a natural process along.
Okay, so why are these organoids such a massive game changer? Well, for decades, research has relied on these flat 2D cell cultures in a petri dish or on animal models. But here's the thing, 2D models can't capture the brain's complex, tangled architecture. And let's face it, a mouse brain just isn't a human brain. Brain organoids give us a human-specific 3D model that lets us watch the intricate dance of cell-to-cell interactions in a developing brain like we've never been able to before. And this powerful new way of modeling the brain, it isn't just some academic exercise. It is sparking a full-blown revolution in medicine. It's allowing us to study diseases and test potential cures in ways we could have only dreamed of just a short time ago.
And the need for this revolution is just staggering. Neurological diseases affect an estimated 43% of the entire global population. Think about that. It makes them a leading cause of disability and death worldwide. This is exactly why this research is so urgent. Using these brain organoids, researchers are now creating models for some of our most devastating conditions: Alzheimer's, Parkinson's, and neurodevelopmental disorders like autism. They can even model the effects of a traumatic brain injury or a stroke, all in the search for new ways to help the brain heal itself.
This technology also unlocks the incredible power of personalized medicine. So imagine this. A patient has a rare genetic marker for Alzheimer's. Scientists can take a few of their skin cells, grow an organoid with that person's exact genetic code, and then test hundreds of potential drugs on it to see which one works best, all before that patient ever has to take a single pill. It's a completely new era for drug screening.
And beyond all the amazing scientific benefits, there's a huge ethical advantage here too. Organoids offer us a path away from animal testing. By using more accurate, human-based models, not only do we get better data, but we can also reduce our reliance on animal research, which is a goal that so many people are striving for.
But the applications don't stop with medicine. Nope. The next frontier is something that sounds like it's been ripped from the pages of a sci-fi novel: biocomputing. Researchers are now seriously exploring how to harness the computational power of living brain tissue itself. This has actually given rise to a whole new field called organoid intelligence, or OI. The ambition here is to use these 3D brain organoids as living processors. By tapping into their natural ability to form connections and process information, scientists believe we could create a fundamentally new kind of computer. So, how is this different from the artificial intelligence we hear about every single day? Well, you can think of it like this. AI tries to make silicon-based computers more brain-like. OI, on the other hand, tries to make biological brain cultures more computer-like. And because they're biological, they are potentially far more energy efficient than our current power-hungry computer systems. The potential here is just enormous. OI-based biocomputing could lead to systems that make decisions faster, are incredibly efficient with data and energy, and can learn continuously while they're working. This wouldn't just be a small improvement. It could fundamentally change how we solve complex problems in fields from logistics to climate modeling. Of course, growing human brain tissue in a lab doesn't just raise scientific possibilities,
it raises some really profound ethical questions. And if we want to move forward responsibly, we have to navigate a very, very complex ethical landscape.
And here it is, the question that really captures everyone's attention. As these organoids get more and more sophisticated, could they ever become sentient? Could they develop consciousness? It's a question that scientists and ethicists are taking very, very seriously.
So to answer that, we have to be really clear about what we even mean by consciousness. Scientists like to break it down. Could they feel things like pain or pleasure? That's called sentience. Right now, it's considered very unlikely. What about having internal subjective experiences, like seeing the color red in your mind's eye? That's highly speculative. The kind of cognitive processing you'd need for complex thought just isn't there. So while they might respond to stimuli, it's more like a reflex than a conscious experience.
In fact, as this quote from the Asia-Pacific Neuroethics Working Group points out, the whole discussion can get clouded by sensationalism. The experts agree that while we have to be thinking about the future, the fears about conscious mini-brains in a dish are, at this stage, pretty misplaced. But that does not mean there aren't real ethical challenges to tackle. Scientists are actively debating some critical questions, things like donor consent. How are a person's cells going to be used in the future? What about the privacy of the genetic data? Could this tech be used for harm? And even the really fundamental legal status of an organoid. Is it just human tissue, or is it something entirely new? So, to address these big challenges, the global scientific community is trying to be proactive, to build a path forward together. The whole goal is to create a solid framework that makes sure this incredibly powerful technology is developed responsibly.
And the guiding principle is pretty simple, really. It's all about balance. Everyone agrees we need to promote this incredible scientific progress, but it has to, absolutely has to, go hand in hand with rigorous and thoughtful ethical oversight.
So what does that actually look like in practice? Well, there are some really concrete recommendations on the table. It means basing these discussions on scientific evidence, not on sci-fi hype. It means developing ethical rules before we need them. It also means setting up crystal clear standards for consent, communicating responsibly with the public, and making sure this conversation includes everyone: scientists, ethicists, policymakers, and all of us. Brain organoids offer these just unprecedented opportunities to understand our own minds, to heal devastating diseases, and maybe even to redefine what a computer is. The journey is really just beginning, and as we continue to unlock the brain's deepest secrets, we're faced with this profound question: how do we, together, define the boundaries of our own creation?